Right, good evening, everyone. So, welcome to all of you to today's session about the Talking 3R Science webinar. And I'm very, very happy today. We're going to talk about one topic that myself I'm enjoying and working in a lot. So, I'm very happy to hear the panelists discuss today about organ-on-a-chip technology, the current state, and the future opportunities. And without further ado, I want to introduce the panelists for today. I have the pleasure of welcoming Kimberly Homan, Senior Director of the Complex In Vitro Systems Group at Genentech, Sonia Bacon from the Federal Agency for Medicine and Health Products in Belgium, and Adrian Rowe, a Principal Scientific Director for Personalized Healthcare Safety at Roche in Basel. Welcome to the three of you. I'm very much looking forward to talking to you and discussing a number of questions. But before we start with that, I want to first ask the three of you to briefly introduce yourselves and give a little bit more of a background about where you're coming from and how you came into the pleasure of working with organ-on-a-chip technology. So, Kim, you want to start?
Yeah, so I have a pretty varied background. In between my bachelor's and PhD, I was in the Marine Corps for 6 years, as you can see here. During my graduate work in biomedical engineering, I helped found a company called Nanohybrids based on a lot of nanotech that I was working on at the time. I also spent quite a bit of time inside a company called Senda, which was also funded by Flagship Ventures in the Boston area. And I spent time being both a postdoc at Harvard and co-appointed at Roche, actually with Adrian's group, working on kidney organ-on-a-chip technologies and also vascular on-a-chip technologies. And then took all of that to Genentech, where I now lead a group on organoids and organs on chip and enhancing our translational potential using those technologies.
Okay, thank you, Kim. Sonia.
Yes, hello everybody. So I have quite a different background. I am a graduated PhD in pharmaceutical sciences and more particularly in vitro toxicology from the Free University of Brussels. And I had also a master in applied toxicology from the University of Surrey in UK. And after my PhD, where I was already active in the Belgian platform for alternatives to animal testing, I moved to the Federal Agency for Medicines and Health Products, where I became the coordinator of the non-clinical reviewers, people that are responsible of reviewing applications for clinical trials, handling applications for marketing authorizations for human medicines, but also giving scientific advice at the national as well as at the European level to drug development companies as well as spin-offs and academic centers. Since the beginning, I've been engaged, participating in the discussions at the European Medicines Agency level, being part of the Safety Working Party, but also being part, and more specifically relevant for today, in fact, of the 3Rs working group that was set up in 2011. And I was chairing that group since then, and this has now become a standing working party. With regards to organ on chip and microphysiological systems, I started, in fact, being engaged from the very early stages of the DARPA project, since AIMB and NIH asked me to participate in their discussions very early on qualification criteria for these kinds of technologies. So next slide, please. And just to give you a flavor of what we are doing currently with the EMA 3Rs Working Party, we are really putting everything in place in order to gain or to facilitate regulatory acceptance of these technologies with a setup of multi-stakeholder workshops to discuss in an interactive manner with all the people involved of how to advance these technologies towards the regulatory field. Later on, we're developing specific guidance, and we will probably come back in the discussion later on performance standards and so on. we do not want to do this alone because, of course, we are in a global sector, and therefore there is a need for consultation with our fellow regulators around the globe to achieve harmonization in acceptance criteria. And finally, also internally, we need to work with the academic centers, and that's why we created the so-called ESSEC, or European Specialized Expert Community, to have a pool of expertise that is joining academic expertise with regulatory expertise and exchange on this topic. And we set up specific drafting groups, of course, for all these regulatory guidances. And further on, we've set up an open entry port for these technology developers and users to engage into a free of charge dialogue with the regulators at the EMA. And this is called the EMA Innovation Task Force, where you can already start discussing whether MPS technology would be suitable to include in the regulations, or whether we would need more data towards qualification and so on. So this is just, in a nutshell, very quickly, a flavor of what we are doing in this respect. Thanks.
This is very exciting, Sonia. So last but not least, Adrian.
Yeah, thanks, Peter. Hi, everyone. Adrian Roth here. I work for Roche, and I'm based in Basel, Switzerland. Originally, I have a molecular biology background, and quite some years ago, this is actually what gave me a job at Roche. So doing genomics, helping introducing genomics for preclinical safety assessment. But shortly later on, I was given the opportunity to form a group that would focus on cellular tools supporting preclinical safety. And that coincided with the advent of the first publications of 3D models and organ chip type of tools from some of the universities. I also just happened to meet then people that were at that time forming the DARPA initiative and joined the Wyss Institute and so on and so forth. So we decided to also then with my team to start looking into that and whether we could apply those tools in support of preclinical drug safety assessment. We're really inspired by some of these findings that we generated. Of course, we also had to learn the hard way that some of these tools were not ready yet for prime time. But nevertheless, we continued, and so also the role grew. And before leaving that role, I was in charge of all preclinical in vitro safety and DMPK activities for Roche's European-based early research organization. Then 3 years ago, I decided to learn something new and joined our clinical development organization, where I'm now part of a group that's looking into personalizing safety assessment, supporting clinical development. And what I bring into this group is also assessing and exploring whether we could use, again, human tissue models, in this case, including also patient-derived tissue models to optimize the safe and effective use of our drug candidates and looking into an individual level. And maybe also to kick off the discussion, I was asked to make a statement about my view on the current state of this approach. So I think we witnessed a lot of encouraging examples over the past decades, but certainly there's still some way to go if we really want to have significant impact on the 3 Rs, and that has to do with things like scalability, allow better reproducibility, looking into really those aspects around biology that are important for our portfolios, and maybe also broadening a bit the type of available models, not limited to the usual ones where with, in some cases, I would say, well covered. And sometimes also what is lacking is a bit a better demonstration. What's really the added value here, and what would be the price to pay for that in order to get there? and so that we can really then move forward and address really a gap and maybe even replace some of those existing animal tests and not just add these sometimes expensive approaches on top. And certainly also it would be desirable so that these many different technical setups, that there's a bit of a way to compare them. There's interchangeability and some flexibility. So just some thoughts that maybe help the discussion. And with that, I think I hand back to the moderator.
Thank you, Adrian, very much. And Kim and Sonia as well. As you might see, if you carefully looked at the announcement, we're actually missing one further panel member. Andries van der Merwe was unfortunately had a family emergency and hence could not join today. So we unfortunately have to discuss without him, but I'm sure we can answer many of the questions as well. So before we get to the questions, and now we have received a nice overview of who is here on the panel, but to get us started, we also would love to hear a little bit from you in the audience. What is the experience you have with organ-on-a-chip models? So you should see a poll coming up right now. And also, would you need to utilize organ-on-a-chip models? Okay, there we are. So as we can see, we have a highly interested audience. We only have about 4% model developers and about 1% industry users, 4% academia users, 1% of individuals assessing data from models, and 72% that are highly interested but don't have any experience, and 17% of the audience that have no experience and don't know yet if it's relevant for them. And I think the second, we can see a clear winner, what is needed, and the clear winner is training. Not many are the opinion they got everything, and training and infrastructure, and also more reference data. That's what 30% say, and 22% say that we need better, more advanced models. So that's, I think, a good starting point, and I'm glad to have a lot of highly interested people here. And I want to get started with actually the first question. After Adrian set the scene already a little bit with his current state, I want to go to Kim and ask you, can you describe in 5 words what an organ-on-a-chip is?
Sure. Cells on a microfluidic thumb drive.
All right. Okay. And I can also give you a couple more words.
Yeah.
What are the, in a more comprehensive, what defines an organ-on-a-chip?
Sure. For me, you could take cells, they could be human, but they could also be animal. And you'd put them in a microenvironment that much better mimics the in vivo microenvironment. So these little microfluidic chips allow you to add stretch or perfusion, cues that are important for the differentiation and maturation of that tissue so that it better mimics the in vivo counterpart. So it allows us to make cells behave much more like they do in the body.
Adrian, Sonia, anything to add to that? Does that fit the definition you also got on with? Very good. I'll hand back to Adrian. You mentioned that you have been using organ-on-a-chip models since actually quite a long time. You were one of the pioneers of adopting this technology in industry. What is the motivation of industry to use these organ-on-a-chip models? So where does the interest come from?
Well, I would say twofold. I mean, first of all, being able to use human cells in a physiologically more relevant environment has the potential that also the data you generate in such a model is more human relevant, and that's something we're struggling with. We know that our models are not perfect, and so that's why not everything turns out to be working as great as we hoped during preclinical discovery research. And so the vision is by using a fully human-based system that we could become more human relevant. And the second important aspect and motivation here is, of course, to be able to reduce animal testing today or in the future to as much as possible. So that was certainly the starting point. Yeah.
Kim, how about your Genentech motivation?
I would say it's similar. I guess just one little flavor I would add is, we're really hoping that these systems can also help us do better translation when we do actually run animal studies. So if you ran, for instance, a kidney organ on a chip for a rat system and a human system, and we know that the rat model, when you put it in vivo, say, overpredicts the toxicity coming from antibiotics, which is known. And then we know that it's going to be scaled down for the human, but how much? So this in vitro to in vivo translation can also be enhanced by using these, organ on chip systems. So in vivo to animal and in vivo to human data that we already have.
All right, so we see there's quite some motivation in different type of application scenarios in industry, especially now talking about pharmaceutical industry. Sonia, you are the one that actually then sees this data. So how much data do regulators actually see from organ on a chip or more general MPS model? And then maybe in the second part already, what is holding companies back, you think, that to submit more data from these kind of models?
Yeah, your second question already responds a little bit the first one. And I can only confirm we are seeing very little data coming in right now within applications for products, so clinical trial applications and marketing authorization applications. Although there are some examples of models being used to establish proof of concepts or to demonstrate efficacy of new medicinal products that are being developed. There are examples out there. However, what we are seeing is method developers coming to us with these methods through the Innovation Task Force to ask about the possible worth of the method within the regulatory process and to see how we could move it further towards acceptance of the data that are generated within these models for a particular context of use. And this will be a terminology that we will be using probably more often in this conversation. And hence also to develop guidance to kind of accept these methodologies. So what are the hurdles? I think what we had as an impression from our workshop that we organized already in 2017 and from later conversations was it was twofold. There is a cry out for more guidance to see what would be the criteria for regulatory authorities to actually accept the data coming from these models. And on the other hand, there is a bit of the hurdle of submitting these data within dossiers as what happens if a signal is contradictory to what you see to the animal studies, and could that stop the development process, or could that slow down the smooth clinical trial follow-up and also the marketing authorization application. So in that sense, we also need to engage into a conversation to allow for a safe space within the regulatory domain in order to be able to look at the data without them being taken into account for a decision making on a product itself, the so-called safe harbor. So these are two big hurdles I think from our side we perceive to be there.
Can I ask Sonia, how's it going with Safe Harbor?
It's there. Everything is in place to set it up, but actually we have had up to now no real requests to start this up. So there have been examples of safe harbors without them being called that way in the ICH space, for instance, where they assessed the weight of evidence approach for carcinogenicity waivers. But in the European Medicines Agency for specific methods, we have not had that until now, although it's clear that if people would be coming for qualification for a method, we could set up such a scheme in order to generate more data to increase the evidence of the use or the worth of the method to qualification.
We already have some questions from the audience, and I wanna briefly include one of them because you mentioned about these stakeholder workshops, and there's a question from one of the attendees. how does my company that is currently developing these methodologies get involved in the stakeholder workshops to influence and support developing more appropriate and translational methods for efficacy and safety assessment of medicinal products?
Yeah, we will be starting with 2 workshops next year. And one of them will be a workshop on liver-on-chip to use within the context of predicting drug-induced liver injury. And the other one will be on the topic of heart-on-chip to predict safety pharmacology endpoints within the realm of safety pharmacology testing. So when these workshops will be set up, we will reach out to the community and to the major players in the field to engage into these workshops and to participate to the discussion. Of course, we cannot get the whole world on board, but we also have another possibility. This is the Innovation Task Force to have a bilateral dialogue with the method developer. This is one side, and on the other hand, we have also, every time we publish something, it's put in the public space, and people have the opportunity to provide comments, input, and this could generate follow-up workshops as well to further on discuss.
Sorry, one thing to add there, Peter, if because there's a bunch in the US too. So the North American 3Rs Collaborative is something that you can join as a startup company in that space. You can also look at engaging with an organization called the IQ-MPS, which is a collaboration of different pharma companies that I'm actually the chair, will be the chair soon of. And that organization has, a bunch of pharma companies that are very interested in this, and they also do webinars and other engagement activities. So both the website of the 3Rs American Collaborative, or not American, NA3Rs Collaborative and IQ-MPS are areas you can look to.
Very nice. And also on a European level, we have the European Organ-on-a-Chip Society, which is also doing a lot of workshops and conferences and has working groups, especially for this type of questions. So everyone interested also, I encourage you to look what is going on there.
Yeah. And Peter, if I may interact, this European Society for Organ-on-Chip has a regulatory advisory board, of which I'm also part, and some other colleagues from the EMA working parties, and hence there is a close link with the regulatory community, not only for pharmaceutical products, but also for chemicals, and EFSA is on board as well.
So there's a lot of opportunities to get involved. And I think this is also key, and this is the communication between stakeholders is one of the key aspects necessary. And there's also a question from Barney Reed on a little in that direction. So how can we bridge the gap between everyone working in academia, developing organ-on-chip technologies, and those in industry and regulatory bodies who might use them or the data coming from them? And how can we better or best ensure that what is being developed is what is needed or what is wanted?
I think this is absolutely key. The collaboration between all stakeholders, and if we say multi-stakeholder workshops, that's exactly what we mean. We want to bring the science together with the method developers, together with the end users and the regulators, because it's really important to, from the beginning of the development, already gear the method development towards the useful tools that will respond the questions that we need to be responded later on in the regulatory space, I think.
And maybe to add on that, I mean, I fully agree, but would also like to say that I in all those years, I can't think of a single model development exercise where we did not work together closely with an academic or a partner or a startup company in doing so. So this is, I think it's the only way to go to work in close collaboration. Certainly then the loop to regulatory is a different one. That's not something that we typically would do right away as part of the model development. Maybe that changes. I think it depends a bit what's the purpose of a model, what kind of decision are you driving. And also, even if that doesn't take place, that wouldn't make such an assay or a model less important. That's also something I wanted to add to the discussion before. There's a whole bunch of activities done that will not be part of the regulatory package. That doesn't mean that they're less important, not at all. I mean, it's what is part of the regulatory package is what's asked for, what's defined. But sometimes all the preparatory essays you do, which guide you in the right direction, they might be even more important, because this is where you decide internally, and in the end you run the essay that's required. And in many cases, you know it will be positive. Otherwise, you're not going to submit it. So just as a comment about. So that there's not the misconception that something that is not part of a regular integral part of a regulatory package is not important. That's certainly not true.
Can I comment there? Yeah, because right now we are kind of making the real distinction between the early phases of screening and discovery, where all the nice fancy tools are being used, and then the regulatory package. And when we're discussing 3Rs and reducing and replacing animal experimentation, it's true that we will not be able just to replace one animal experiment by a multi-organ on chip just from here to tomorrow. And I think what we need to evolve to is the integration of results coming from these innovative tools that are being used very early and in a decision-making scheme where you will decide on the basis of the evidence that you generate, not only for MPS, but from different tools, what kind of studies will you really need to answer the questions that you need to answer? And hence, I think the cooperation with the regulatory authorities in method development is really crucial to really help that kind of shift towards this new thinking, and to see what kind of information could we accept and do we need to put into such a weight of evidence approach. Because if you look at the current regulatory evolutions in the guidelines, weight of evidence is everywhere, and it's really important that we feed this weight of evidence with relevant data, and tools like this could provide really relevant data.
And one quick thing to add to those wonderful comments. So the internal decision-making is where most of the models are being used right now. But I'd also like to add from a 3Rs perspective that we're also trying to use the models to justify doing either not two species or not doing cynos. So when we think about how to even do the studies that might be required, we're trying to amend them or change them based on species selection that we can do from in vitro modeling and from weight of evidence approaches.
Yeah, I think this is a very, very important point. Let's say the range of where the models can have an impact is quite wide, even in pharmaceutical industry. I don't want to focus only on pharmaceutical industry, although we are heavily focused on that here in a panel, and we lost with Andres, our academic member. But I actually want to have a question. Maybe Adrian, you can help out here. I mean, the non-clinical research in pharmaceutical companies has been the driver of, or one of the major driver for organ-on-a-chip adoption. You yourself drove a lot of that at Roche, and now you switch more to the clinical part. And so the clinical research, but also the academic basic research has been lagging strongly in kind of using or taking up these technologies. What do you think is necessary to see more adoption in the areas of clinical research, both in pharma, but also in the academia, and also academic basic research?
Maybe I'll try to answer the second one first, because I guess that's to me the easier one. I would say that I mean, what has hindering academic groups of adopting the technology, I would say the price was an important driver, especially early days models, have been sometimes, not all of them, have been very, very expensive, and that's just beyond the budget of a normal academic group. Plus, then depending on the area of research you're in, there's certain, let's say, norms, what is required for in your field for a high-end publication. And in some fields, it has remained the knockout mouse model or whatever it is. And that is something that may require quite some time until it changes, including then also manuscript reviewers and journal editors, especially those from high-end journals, to really also support such a change in mindset and also look into such alternative models. Maybe colleagues have other views, but that's what I would say. Now to the second question, the adoption in clinical research. I mean, I'd say that's just something which is not that is relatively new. Yeah. In this case, I'd say it's actually university hospitals, big clinical research organizations that are clearly at the forefront and driving it. I know of a number of large clinical organizations that are running, as we speak today, clinical trials with such models being included that drive treatment decisions. Here in pharma, that's certainly something new that people find interesting, but want to see more data, and I hope I can contribute there soon with some of the first proof of concept data that this can also help in the clinical space.
All right. Any thoughts from Kim or Sonia?
I mean, the price is still, the price is still a driver for us too. So even though I mean, I know most of the academic groups working on this technology will make their own just so that they can, reuse parts or do it cheaply. And just for an idea for people who, because a lot of people on this call are interested in this field, some organ-on-chip technologies cost let's say $600 for an N of 1, and like a mouse would cost $60. So with a mouse, you would get 50 plus organs you could analyze, and with an organ on a chip, you're going to get one part of one organ for an order of magnitude more cost in some cases. So the cost is really a driver here, and I do see a lot of movement in the startup space where companies are going to higher throughput, and we're looking at all of those. And I would definitely say that we have to push in that direction, and hopefully that will change in the near future. But it is definitely a driver, as Adrian mentioned.
Yes, and I think this is also, I wanted to come to the costs because there is a good question from the audience. So in general, put all your questions you have in the Q&A. But there is exactly a question on the cost, and I think, Kim, you have already answered that almost. But the question from Angela Ramirez Valades is if we could talk about the cost and how much an organ-on-chip costs, and if this could be a solution to economic limitations that biomedical research faces in emerging countries. And I think this is, partially you already answered it, Kim. Maybe you can get a little bit more into detail in this. What is the range we can see? But maybe I want to add one thing to that. How can we, let's say, ensure that this technology is not only taken up in, let's say, the wealthy nations, but that this technology can also be used in, let's say, countries or emerging countries?
That's a hard one.
Mm-hmm.
That's a hard one. We, from our side, if we start adopting these technologies into our guidelines, it will certainly be immediately at hopefully we can get them at an international level through ICH or VICH, although it's clear that it will be a stepwise approach. Once it's there, we have a broad representation of different regions in that conversation, and this could help adoption in other countries as well. Linked to the price, that's of course another discussion.
I guess I could try to add, this is really hard to answer, so right off the bat, I'll agree with Sonia there. But I think that there's a recognition generally that pharma needs to do better at inclusivity in our trials and inclusivity in our preclinical studies. So as a recognition of this need to serve, more of our patients by actually checking multiple cell lines of different genetic ancestries of different sex. One of the initiatives that we have in Genentech is to reach out to many different countries or other places that we have not usually engaged with. And both, we're doing things like trying to do technology exchange and information exchange with these places. And in exchange for that, we're getting cells from their native populations so that we can use them in safety testing and be more inclusive in our preclinical studies. So for instance, we might not catch, liver toxicity from a certain population with a certain CYP that we just don't get from a European or American cohort, for instance. But if we caught it and had it from their population, we could account for it in our studies, for instance. And so there are some instances where we're aligning with countries that we wouldn't usually talk to, and then figuring out how do we bring those countries up? For instance, how can we bring people from those countries to our labs, train them to do particular cell isolations, or understand how to do multi-omics on some of their tissues? How do they take that knowledge back to their home countries and use it effectively? So we don't have a solution, like a full solution, but we're trying to be part of, bringing others up with this technology, if that helps at all.
Any thoughts on the cost? Because I mean, Kim, you made a let's say let's call it a grave example of how much an organ-on-chip experiment can cost. It actually does not have to. I think this is the key from a model developer and also from the fabrication side. The more uptake we have, the more scale up in terms of fabrication you have, the cheaper these products can be produced. So in the end, the most costly factors of most of the models that we are using and also we are working is often the cell. So the cells themselves have if you buy a quality controlled cells, can make the major cost factor there. So the cost can and will go down in the future. I think this is the key.
Yeah, I like your point there, and that's also what I would bring in here. I mean, on the kind of the hardware parts, they are expensive, and maybe for other countries, one approach could be to make these reusable. I'm sure that's technically doable. The other thing would be if countries, that would like to, foster their own MPS organ chip type of research, that they ease donation of cells to be used from within their country, because that's, as you said, that's actually a cost driver. I mean, that's in for some of these organ chip models, it's by far the most expensive part is getting the cells, because we have to purchase them from commercial sources. So that could also be an opportunity for such countries if they could sort of organize that from within their country, so that this is not like the business of a single vendor that then can dictate high prices.
That's true, Adrian. And I also want to just emphasize that, the ethical concerns with that really vary via country. Some people feel that giving their cells is giving away a piece of them, and that is a real thing, a religious thing. So I think that we just need to acknowledge that there would have to be understanding. We have to do a lot of education, patient consent, and all of those things. But I think that things like this, us talking to everyone and making sure the word gets out about why we're using it and how we're using it to be more safe, using it to get better translational outcomes for all patients, and making people understand those messages, I think would be important for us as a community.
Yeah, that's really an important point that you raised there. Training and communication, I think that's really key. And here also in this discussion, I think training all the communities involved is really key for progress.
I think it's a very important topic. Now I want to shift a little bit also more to the technology. We said it's current state and future opportunities. So from a technological side, and now I'm looking at Kim, you have this kind of this unique position of being a model developer, but also an end user. So you have a very broad and good overview there. So where do you think technologically are the major challenges the technology has, and also where are the opportunities that we can get to by addressing these challenges?
I guess I'll start with the cell challenges. They were already mentioned. But even once you have the cells in hand, in different people's hands, they'll grow differently. So for instance, if one person takes an organoid and then wants to put it on an organ-on-chip device, and all of the things that they do beforehand might be different in terms of like quality control, different people looking at the cells and determining they're healthy, they're not healthy, or they're ready, they're not ready, to take them onto an organ-on-chip device, for instance. And so some of the things internally that we're working on at Genentech is applying a hands-off approach, so an automated culture system to these things that also integrates, for instance, AI ML techniques to take pictures of the cells and have the computer tell us that they're good, they're not good, they're ready, they're not ready, so that we're having a more generalized approach across many groups that could work in terms of having standardization of the cultures themselves. And then on the technology development, I think, having perfused, for instance, organ-on-chip devices, at scale is quite the thing, right? So individually controlling flow rates for each of these is really an engineering, technological challenge. And I've seen both academics, 3D printing companies, startups really are working on the development of these, but how do we democratize the use of those, I think, is going to be the future. So how do those companies actually propagate those devices? Do they just stay as a CRO that, you send your compounds or your questions to, or do they sell the devices themselves? And I think that's kind of where the bifurcation might lie for a lot of people who are thinking of developing these technologies. But I would really encourage people to think about scale and how to package down that cost so that we can all benefit.
Adrian, anything to add from your side there?
I fully agree to everything Kim has already mentioned. That was, I'm just thinking of anything on top. Not right now. I think these are really the important points. We made similar experiences regarding the cells, that can behave differently in different people's hands. Maybe on top of that, what we also observed is that, the same cells being maybe used in slightly different models developed by different groups for a different purpose. It seemed like the model has been optimized so strongly in order to meet the criteria, so that it becomes an attractive assay, so that it's almost not physiological anymore. And when you use it in a different, you see that the same cells would behave totally different. Or you measure something else that you're interested from those cells, and you realize these are not physiological conditions anymore. It might serve that particular purpose of assaying endpoint XYZ, but T cells are clearly not in their physiological state anymore. So that's also something we observed, because there's obviously competition, and there's eagerness, to have the best model in place to address a particular question, and that sometimes leads to assay conditions that are not physiological anymore. Maybe just as an additional example.
And how about the let's say the application areas, also the tissues and organ functions? So I think one of the really upcoming challenges, but also huge opportunities, is the inclusion of aspects of the immune system, where you see more and more now coming out. But also one of the biggest challenge from a model developer, especially talking about the cells, allogenic, autologous cell compositions and all of that, but also one of the biggest opportunities. So how do you see that in that direction?
Let's go first. Highly desirable, yes. At the same time, probably also have to be realistic what's doable. To have like a normal organ without the immune component represented there can mean a lot of work and refinement until you really have something where you rely on, where you think, okay, I can use this model to address a specific question. Immune biology is super important. It's integral part of almost every drug development program we have in place. And so naturally, we're super interested to have models that represent certain aspects of the immune system. at the same time, yeah, this will not be realizable tomorrow. Certain aspects are just inherently difficult. But we see already encouraging examples. I think also things like both stem cell CRISPR technologies might be really useful there, at least to solve certain problems, so that we can use immune competent cells in these systems.
Maybe Sonia, from a regulatory standpoint, where do you see? I mean, you already kind of touched upon it, but where do you see the big challenges ahead?
We need to communicate better. There is a lot of conversation about validation of these methods, context of use, standardization, qualification, and it's not completely clear what the expectations are. And when you're engaging with the regulatory authorities, such as our agency or the EMA, we talk about context of use qualification. So that means that for the model that you have before you have already thought about the application in the drug development space, and you want to seek approval for the use of this method in that space. So often in these discussions around 3Rs methods, and that's not only for MPS like that, it's for all type of methods, we see from our side a drive to always optimize methods, to make it better, more physiological and more human-like, etc. And at a certain point in time, you have to stop and say, okay, we have still a model, and the model will always have limitations, and we can live with them. But we need to acknowledge what can we use the model for and what can't we use it. And that's exactly the purpose of such a context of use qualification. If you want to use it for the purpose of predicting liver toxicity, what will your model be able to show us? Even without this immune component on board, what will it be able to do and what won't it be able to do? And what is the place that you can give that method in the decision-making process? And to flesh that out, it's kind of a bumpy road, let's say, and to see what kind of expectations do we have for particular context of use, such as predicting DILI might be one, but using the method in the context of predicting efficacy of a drug, your requirements might be, it's a low-hanging fruit. You can already submit data obtained with these methods for clinical trial application or for a marketing authorization application, because a lot of the in vitro tools are already being submitted to the authorities. It's very research space. So that might be a direct road to submit data, to use data, to get the regulators also acquainted with these models. The safety aspect is something else, and there we need to also this dialogue. So there is a question of terminology and of expectations from both sides, I guess, that need to be clarified. And then the next step is to really to develop this guidance, because often you hear, well, we need to recommend a model, but we are moving away from that space of recommending or qualifying or validating just one method. We will give you the criteria under which we will accept the data, but the technological process and the science is going so fast that we will have to have these flexible criteria to accommodate for all the novel methods that will come along. So that's another challenge for us and for the method developers out there as well.
Maybe Adrian and Kim, on that point also, how do you decide what model to use? And I think this could also be something which is very, very, let's say, tough to do for an academic user who might not have the, let's say, financial power to just test a number of models. But how do you decide what's the model that's going to answer your question?
Yeah, I'll start here because I have a very passionate answer to this question, which is, so when Adrian and I started in this field a long time ago, there might only have been one model, built to answer a particular question. And now there's a plethora of models out there that could answer your question. And so Adrian was alluding to it earlier of, how would you choose between these models? And it's like going to the grocery store where if you had 3 cereals to pick from, like, oh, I like this one best, but now there's a whole line of cereals, like how do you pick which cereal? So in pharma, the way I think about it is that I would love for the community to create what I call model omics. So meaning if I took the same cells, I put them on a transwell, or I put them in an organ chip, or I kept them in a 3D culture in a dome, for instance, like I need to know the biology I'm buying by going to a more complex system. So what fundamental biology changes across those things? And to get that, I would really highly recommend that when you're creating your models, you don't just show with one drug and one proof of concept experiment that it would have predicted the human result. Instead, I would much rather see that there's transcriptomic data across multiple model systems. And now I know, for instance, at Genentech, now I can go mine that data and I can ask for my new drug with this receptor and this engagement pathway, I have to go to this model for this question, right? And so it becomes really, really necessary that if you do bulk RNA-seq, or you do single cell, or you do any proteomics or glycomics or any of that technology, that you publish it with your paper so that I can then take that data into our, ingest it, and then I can look across the model systems and really make an intelligent decision about which model to select for which question. You could see a paper I wrote on this if you'd like. It's in Advanced Biology because I've been very, very passionate about this topic.
Yeah, I can only echo that. I like the idea of the multiomics analysis that could help, explore, how about my current use case or context of use that I'm interested in, because most of the, say, liver models, they have been characterized using acetaminophen. And that's fine, then it detects acetaminophen or take glitazone toxicity. And then you can conclude, okay, it's better than whatever 2D cells or something like that. But maybe my context of use is a very different one. And this can go in both ways. I can maybe a very simple cell line might be absolutely doing the job. And it's sometimes indeed a very difficult decision, what is it that we're looking for, and does the system we're interested in, does it have the capability to address these questions? And yeah, in early days there was oftentimes not a choice. Now it's oftentimes too much of a choice, and it's difficult to judge the validation data that you get for some of these models to be able to show, is there really something in there, or is this just a me too model, and could it really be superior? That's very difficult to judge for us.
But for us, that's also a crucial question. If you say it has been shown with just one drug like acetaminophen, that won't be sufficient. We really need a set of compounds to agree upon, almost per context of use, to be tested as a kind of baseline to show that your model is predicting at least the basics. And to open the possibility to add compounds to enlarge your space you're interested in, or as a company, the space you're developing drugs in. And if you look to the example of the ICHS5, so that's my passionate space, where we developed guidance for reproductive toxicology, and we kind of elaborated all the qualification criteria you needed to adhere to accept alternative models for embryo-fetal development testing. We agreed on a set of reference compounds, but this was a very curated list where we really needed data from the animal experimentation, human data, and to build that list, that was a whole endeavor. So if we are embarking now into the MPS space with a different context of use, we will have, I think, very long discussions on reference compounds. But it's really important that we have this kind of curated list, because there are a lot of reference compound lists out there, but I always lack the underlying data, and have they all been curated in the same way and looked at in the same way. And I think that's crucial also, to have this baseline agreement.
I'm looking at the time, and I think we could continue this discussion for at least another hour. But unfortunately, we already had almost at 6 p.m., at least in Europe, in Central Europe. So I think we can see there has been a lot of development, and there's a lot of opportunities, but there's also a lot of challenges still ahead. And I would really like to thank all three of you. So thank you, Kim, Sonia, and Adrian, for all these valuable thoughts and this discussion. To all participants that attended, I hope you learned some new things about organ-on-a-chip technology, and maybe you now know how it could be of use for you. Thank you very much.
Thank you.
Thank
Bye-bye.
you. Take care. Bye.